Okay, in this audio lecture, we're going to be looking at measuring microbes in the food. So what different methods can be utilized in order to detect or evaluate if foods, food ingredients, and environments have microbes present in it. So again, I'm looking at these methods. They are used to evaluate and detect microbes in food or food ingredients or the environments in which foods are being processed or maintained in. We can have quantitative assays, methods, and that's going to ask how many, or we have qualitative, and that's are they there or are they not. So for quantitative methods, they're designed so they can actually enumerate or estimate either through direct or indirect methods how much microbes, so what is known as the microbial load, is in the tested material, okay, in the sample that is being tested. So with each method, enumeration or estimation of a specific group among the total that's normally present in the food. Some different examples of these quantitative methods would be total aerobic plate counts, or TAPC, or standard plate count, TPC. So standard plate count is probably one that you did in your intro micro lab. Along with that same kind of premise with standard plate count is anaerobic counts, psychotrophic counts, coliform counts, Staphylococcus aureus counts. With these, we're changing the plates that we're using so that we're counting specifically for a specific type. So the method in which it's performed is slightly different. And then we can also have yeast and mold counts. So different media needs to be used if you're growing yeast and molds, because in many cases, plates that are used to grow bacteria actually have antifungal components to them, like cyclohexamine or several other different chemicals can be added to the plate to inhibit the growth of fungi. So again, you would have to use other methods, other types of media in order to grow the yeast or molds for counts. For qualitative methods, again, we're just trying to determine whether microbes are present or not. Usually a specific microbe is being looked at and whether it's present or not. And many times with qualitative, we're looking to see whether a specific foodborne pathogen is present, because if this is going to cause a high fatality rate, we do want to make sure that this is not going out in our food products. So if it is present, we want to make sure we stop and not release the food that's being produced. So some examples of quantitative methods used to detect specific species would be, looking for the presence of Salmonella, Clostridium, E. coli, especially, again, highly pathogenic strains that produce like Shigella toxin and cause hemorrhaging. Also Listeria. Listeria can cause fatalities, especially in pregnant women. And so again, tests would be done to make sure that food was free of Listeria. When looking at food products for microbial analysis, a lot of times what happens is, you can't test at all, so you have to sample. So what happens is you have a batch or a lot, not like a lot, like it's actually a large amount, but a lot, like if you look in the back of like your Tylenol bottle, it's going to have a lot number, and then everything with the same lot number was produced at the same time. So that's when we say a lot. That's what we're referring to as a batch of food that was produced at the same time. So they can be different states, but we're only going to test a small portion of that, and that tested portion would be known as the sample. And the determination of how you come up with the sampling, a lot of time that is in the standard operation protocols, the protocols that are going to govern what methods you use to test for which organisms. With these different methods, it's important to keep in mind that just because cells are present, just because microbes are present, it doesn't mean that we can culture them. So there are pros when it comes to quantitative methods or conventional methods that require culturing. So some of the newer methods like biosensing and even looking at immunological assays like ELISAs, it doesn't require the cell to actually be cultured. So there's a huge benefit to that. So this term of viable but not culturable is abbreviated VBNC. So in the food microbiology world, that's just like you see that and you know. In some cases, it's called not normally culturable. So NNC. So those are terms that you should know. And there's a whole list of these organisms that, given kind of the normal recommended methods, they aren't detected. You would have to do additional things to either supplement the media to allow them to grow, or again, different techniques where you're just looking for remnants of genetic material would be much more successful in being able to determine that whether they're or not. So I always tell my intro micro students, we can only culture less than 1% of the microbes on this planet. And the same as for with these potential foodborne pathogens and microbes that are present in food, that you have to be able to culture them in order to do some of these techniques. And so that would be a huge downside, but we do have to be aware that they exist. So some of the Vibrios, Salmonella, E. coli, K. pneumonia, Listeria, Pseudomonas, again, these are all, they traditionally are more difficult to culture, like H. pylori. Some of these need just a different pH. So if we think about what we were talking about, factors that influence, we have to have culture media setups, incubator setups to meet those demands. And again, because these are kind of niche bacteria, the standard microlab isn't set up for that. So looking at quantitative and direct measuring or enumeration, we can do microscopic counts. So in this case, in many cases, cells would be stained and then looked at under bright field, or in some cases they could be looked at phase contrast if we were trying to look specifically at like living cells, but some of the stains are stains that are excluded by living cells, so we'd be able to indicate whether they're alive or not, so whether they're viable or not. And then we're able to calculate how many microbes are present per microliter or gram of a food sample. So in the figures you can see an example, the counting chamber that's utilized, so it has a grid, and so when you pipette in your sample, what falls in those boxes is a set volume, so you can then go back and take your counts and calculate the concentration of the microbes that are present. And because you started with a certain volume or certain weight of material, you'd be able to determine whether your microbial counts are too high or too low. And again, each of these, with different food products, there's a certain standard in the number of microbes that are allowed. You can also use fluorescent staining. So here we have a staining that the viable cells are green and the dead cells are red. And so dead cells can't divide. They for the most part, they can't cause infection. They could cause illness. So if you had a high amount of a bacteria that's producing like a toxin or is gram-negative and is releasing LPS, so lipid A, definitely those dead cells could matter. But for the most part, they would be looking at those viable cells because those are the ones that could grow and divide as the food is being stored before it's consumed. Another method of direct enumeration is colony forming units. So this can use different types of media. So you could have non-select media, or you could have non-select but differential media. You could have selective media, or you could have selective differential media. And we'll talk a little bit about what all those terms mean. But basically with colony forming units, again, this is a technique you probably did in intro biolab. You'll do a serial dilution. So you'll take your stock solution, and this could either be your food item or it could be a set amount, like 1 gram, and then you dilute it into a diluent, and then you repeat the same dilution over and over again. So a 1 to 10 dilution, a 1 to 100 dilution, and then you take a small sample of that, and then using the spread plate method, you spread that small sample out onto the plate, and then you incubate it and allow it to grow. So one of the things to keep in mind with the standard plate count, depending on the media you're using, normally there's going to be a set kind of protocol. So for a standard plate count, you're going to incubate that for 32 degrees Celsius for 48 hours. If we're using different media, or we're looking at different types of organisms, like those psychotrophic bacteria, for those counts, they would actually be at 7 degrees Celsius for 10 days, or 10 degrees Celsius for 7 days. So that would be a much longer incubation period than what would be present if we were just doing a standard plate count. So again, protocols would govern how long. So it's not, it's not like the microbiome lab where you're like, okay, well, we don't meet again for a week, so it's going to incubate for a week. You would have to pull plates and do readings at a set time to do that. So if you look at the PowerPoint slides, there is a video on serial dilutions and plate counts. It's actually from Bioreb, and it goes through the process of doing a serial dilution and then how you would do the spread plate method. So please do watch that, especially if it's been a while since you've done that, because you should be familiar with that procedure as it's used for standard plate count and some of the other counting methods. So as I mentioned with the standard plate count, you can use different types of media. So again, just as a refresher, you should know the different types of media and what those terms mean. So general purpose media is going to allow a wide variety of organisms to grow, where a selective media is only going to allow certain types. So the components within the media will actually limit which types of organisms grow. So an example of a selective media would be the mannitol salt agar plate, or MSA plate, you might have used, because it's a more acidic pH for the media. It doesn't allow a lot of those neutrophils to grow, or neutrophilic, immunology in my brain. So it doesn't allow those neutrophilic bacteria to actually grow on the plate. So again, it's those more of those acidophiles or bacteria that's likes a high salt concentration is another component of the mannitol salt agar plate. And the other piece to that plate is that mannitol. So it has to be able to use mannitol as a sugar source. So that kind of limits what can grow on there. With a differential media, again, it can be kind of selective, and you can either have it non-selective or selective, but in many cases it is selective, so not as many grow. But what's unique with the differential is that they change color. So depending upon the organism, if they undergo a certain metabolic pathway and utilize some of the material, they would end up changing color. So an example of this would be the McConkey agar. So the McConkey agar looks at lactulose fermentation, and if the bacteria undergoes lactofermentation and takes up bile salt, it will turn a pink color. Okay, and so you can actually determine the different types of bacteria based on whether they turn pink or they don't turn pink. There's also a number of coliform plates that you might have utilized, where if E. coli grows on it, E. coli has a green sheen to it. So again, that's differential because it lets us know what genus species it is. So just keep in mind that if something's growing on a selective or differential media, some of those colony morphology indicators cannot be utilized because we've altered that environment from what we would normally see, okay? So with a general purpose, things like pigment and color can be seen because we're not adding reagents into the media that cause the bacteria to change color. The color it is its kind of true color. Where if we're growing something on a differential media, the color it is because of the media. So again, we can't really include that as a way to identify the microbes in general, we could only be able to identify them by that color on that differential media. We also have indirect enumeration. So for this, we're not putting them onto the plate and counting. Probably the most common method you would know about is using a spec. So you would take your sample, you do a serial dilution, and then you would take an aliquot, part of that sample, and then read it through a spec. And you're looking at turbidity, so how cloudy is that media, because if the media is cloudy, that equals microbial growth. And so with the spec, with the light source, if the sample is cloudy, you're not going to get as much light to the detector, and so the spec then reads this out. The light has now been absorbed, so you'd have a higher absorbance, where if it wasn't cloudy, if you didn't have as much microbial growth or any microbial growth, more of the light would go to the detector, and you would have a lower absorbance, okay, because the light was not absorbed by the material. Because we have different dilutions, and maybe we have hopefully have a standard with this, we can take that absorbance and then work back and figure out what the concentration of our unknown sample is. So you're not directly counting, it's because of the absorbance reading and using a standard curve that you indicate what the concentration is. Another example of indirect enumeration is the most probable number, and so in this case you use a selective broth. You end up doing a series of dilutions, and you have like repeat, you can see how you have these repeat test tubes, and then they get incubated, and then because it's in this selective broth, it only allows certain organisms to grow, right? And so you're then looking at, okay, are there, do the tubes actually turn positive? Yes, they do. Okay, well then it's most likely, it's probable that the number would fall into this, because we know we did this dilution and this is the response we got. So that one doesn't tend to be as quantitative. It's a little bit more qualitative, just saying, you can definitely say they're there or they're not, but working back to an actual number is not as specific of a number, but there's benefits because you don't have the additional steps of plating out, so you don't need those material. Also, the incubation time tends to be a little bit shorter because you have these higher concentrations, so they're going to react to the media quicker. You don't have to, they don't have to, grow as much. With another example would be the dye reduction test. So there are certain dyes, you probably have heard methylene blue used for staining, and what happens is it has a color to it, but when it gets oxidized, it actually becomes colorless. So we can utilize this property, this color change, to actually determine whether there's microbes present, because microbes will end up reducing the dye if they're present. So this test with methylene blue is routinely used in the milk industry, and so what you'll do is you'll take some of the milk sample, add methylene blue to it, and then you'll see whether it changes color. There's actually a scaling that, depending upon how long it takes to change color, so you'll see in that table there's MBR, which is methylene blue reduction test, and if it ends up changing color within half an hour, that would tell you that the milk is poor quality. And what they've done is kind of link this back to a total plate count, so that if you did a total plate count, you'd have, your calculations would work out to be over 5 million CFUs, colony forming units per ml, where if it takes an hour to 2 hours for the loss of color, then that would indicate it's fair, which means you now have 1 to 5 million CFUs per ml. Obviously what we're achieving for is above that. So hopefully you have a very good sample. So it would take longer than 5 hours for any color change to happen, which means there's not more than 0.2 million colony forming units per ml. So like that most probable number, the dye reduction test, yes, there's kind of this back calculation of total plate counts, the CFUs per ml, but the reality is it's, again, a little bit more qualitative, saying is it, or are there organisms present or not. So if you kind of read it at a certain time and it hasn't changed color, then you can be more confident that there's not enough microbes present to affect the sample. And a lot of times this is done for raw milk before it goes through processing, so that they know how long do they have to process or pasteurize, or do they have to throw this sample batch of milk out and not incorporate it into the overall milk production. So there's also specific tests for bacterial toxins, and these would be quantitative. So is the microbe present or not? Generally, you might look at the concentration of toxins, but this doesn't always relate to how many microbes are present. Okay, so Staphylococcus aureus, Clostridium botulism, Bacillus, and Vibrios and different molds can produce toxins, and that can cause foodborne poisoning. So foodborne poisoning usually refers to toxins being present, not necessarily the actual microbe. So as I mentioned before, microbe could be dead, but if it's already produced toxins and there's toxins present in the sample, it doesn't matter that the bacteria is dead, people can still get sick. Okay, so for the testing of bacterial toxins, it's going to depend upon what toxin you're testing for, which method you utilize. Okay, so there's a number of different methods. Many of these are immunological based. There's a microslide precipitation method, so that utilizes antibodies that would recognize the toxin and clumps them together, so you get this kind of agglutination, similar if you're doing like a blood typing, or if in the micro lab you did the coagulase test. It's kind of similar to that, where you end up getting clumps forming. So if you have clumps forming, the antibodies agglutinated together because they were binding the antigen. What they were binding was the toxin. Other methods would be radio immunoassay, or you're probably more familiar with Enzyme-Linked Immunosorbent assay and ELISA, but we can also do this reverse passive latex agglutination assay. Say that 10 times fast. But there's also biosensor based technology, and we'll talk a little bit more about the biosensor later on. So again, we want to keep in mind that some of the conventional methods that are utilized are, looking under the microscope and trying to detect using either light or electron microscope, culturing methods, and again, those are methods that a lot of them you learned in an intro microbiology lab, which involves screening and selecting and enriching and utilizing, all these different types of media. But they also take time. There's also biochemistry, and this is where we can use some of those selective medias and look at fermentation and what enzymes are present through these selective medias, or by direct detection of enzymes. And then we have immunological based techniques, again, like ELISA or immunofluorescence. And then we have these molecular techniques where we could do genotyping. We could look at the 16S DNA sequence of the ribosome, or we could do PCR or hybridization with different probes. So those are definitely some of the conventional approved methods. A growing area is looking at these biosensors and APTA sensors. So the conventional methods, they can vary from taking a few minutes to 6+ days, depending upon what method you're using. There's also your average worker at your food industry may not be able to do the technique, so there's a lot of additional training, which is great for us as biologists and scientists, but that kind of limits the accessibility of some of these techniques because you need to have devoted lab space for it. With the biosensors, usually within a few minutes or a few hours, you would end up getting results. So they're much quicker. And also because of the way the methods work, a lot of times, as long as they're trained in performing that technique, they're usually kind of like a snap test, or there's a machine component to put the sample in, press run, and get a result. So there's a technical piece to it, but again, they're usually a lot easier to actually conduct than we would look at conventional methods. But there's not as a steep learning curve. So again, some of the pros of traditional culturing methods is that it can be highly sensitive because of the different media we're using and because we can do counts on them. And the media in general tends to be relatively inexpensive, so it's more the personnel that's the expense. And you can do both qualitative and quantitative analysis. The cons of the traditional culturing methods is that it's labor intensive and it's lengthy depending upon the test to get results. And again, if we're talking about a food product, a food product has to be held until it's found to be free of microbes or safe to consume. And if for some reason someone did the test the wrong way, you're potentially looking at having to recall or lawsuits and such. So there would be many cons in addition to that, and a lot of those cons cost money. And so when companies are trying to make their food products affordable and still have profit margins, that's obviously a downside. So the development of rapid methods using metabolic fingerprinting, these immunoassays, using bacteriophages, those viruses that infect bacteria to detect whether the pathogens are present, bioluminescence, nucleic acid-based methods, and biosensors, whether they're pathogen detection or cell-based detectors, have many benefits to them because you get more rapid results, which means food products can be released for consumption, for sale, much quicker. And again, as I mentioned, a lot of these, some of that additional training that's required for culturing methods are not required for these rapid because they're high throughput machines. So it's a little bit. Some of these have actually been robot made, so that robots are capable of doing it. So then that's where the scientists come in on the technical and the oversight, just making sure the equipment is running correctly. So this table from this article that looks at different detection methods for Salmonella outlines some of the advantages, disadvantages, and what progresses have been made for these different areas of detection. So I do highly recommend looking at this. It's some of what I've already have mentioned. Again, really focusing on the cultural colony-based methods, because these are the traditional microbiology methods, ones, again, you would have learned in an intro micro course. So definitely has advantages on the cost of material-wise, but then the drawbacks would be it's very time-consuming. Also, a lot of times it doesn't account for the viable but not culturable aspect. And so as far as progressing that area, looking at association with DNA, antibodies, and chemical-based methods, which kind of leads you into saying, okay, culturing methods aren't really great. Let's look at immunological-based methods. So next column, right? So with immunological based methods, the advantages are you're going to get faster turnaround. You can also run a higher number of material. And because of the fast turnaround, you're almost getting real-time analysis of what's going on. And it can be highly specific. Some of the drawbacks is because of how these immunological-based methods occur. Sometimes they don't have sensitivity. So we'll talk a little bit more about that in our next slide. And also you need antibodies that have good affinity to the pathogen or material from the pathogen, and so that's not always possible. Okay, so this is where things like PCR come in. So PCR is highly sensitive, it's rapid, it can detect whether there's a small amount of nucleic acid present, and so you don't have to have the cells alive. The drawback would be that you can't tell whether the cells are alive or dead. You just know that the genetic material was there, so it may not be present. Okay, so reverse transcriptase, RT-PCR, actually it does allow you to distinguish between live and dead cells. So in many cases that would be the preferred PCR method to use then. But this is where some of those biosensor techniques are probably a little bit better. Okay. So looking at a couple of the different immunological assays, with Enzyme-Linked Immunosorbent assay, you probably did this in a cell bio course, or we even do it sometimes in like an A&P course or an intro bio course. Or if you've taken immunology, this is a technique in which there's several different methods in doing this. So you can see down in the legend, we have an indirect method. So the antibody is recognizing the antigen, and then another antibody comes in and binds, and then you get a color change. There's also a sandwich method. So you can see here where you have a capture antibody that has coated the plate, and the antigen is sandwiched in between that capture and the detecting antibody. In some cases, a third antibody is utilized to bind the detecting to get the color change. And then we can also have a competitive ELISA where you have antibodies competing for binding to the antigen. So I would say that you should know that with an ELISA, there's different formats that can be utilized, and so depending upon what antibodies are available, depending upon how strongly they bind, depending upon your setup, you may use one over the other. So the capture antibody plates have to be labeled with the antibody, so depending upon whether they're using a commercially available ELISA or whether they've developed their own ELISA. Most food industries are going to use commercially available ELISAs. That may be the only method that they have available. With indirect, the plate gets coated with the antigen, and so you have to have special plates for that. So again, there's a whole bunch of different factors, and again, usually in the food industry, they're using commercially available ELISAs that have been standardized, that are approved by the USDA or the FDA in order to be utilized for that. So they're not, in general, unless they're running a kind of research program, they're not developing new ELISAs. So what they purchase is what they would utilize. The benefit of an ELISA is that usually these are in a 96-well format, so you can run a number of samples in triplicates at the same time. Now with the lateral flow assay system, these sometimes are referred to as snap test or dipsticks, and so in this case you put a sample in the area that's labeled sample pad, and then either this could be in a cassette, like they are for the snap test, or this could be just like a dipstick that you use to check glucose, right? And so you put your sample on, what happens is that because of the properties of liquid, it ends up moving towards that other end where the absorbent pad is. And as it moves through, it gets drawn through this conjunctive pad. And so in there, this is where we would have antibodies present. And then it moves towards the test line and control line, which is in this nitrocellulose membrane. And when it reacts to these different substrates, if it's brought antibodies with it, it will react to the material that's in the nitrocellulose membrane. So different substrates are added in there, that test line and the control line. And so again, if the sample has bound to the antibodies, because maybe these antibodies are to Salmonella, and Salmonella has now moved through, or proteins from Salmonella have moved through, when they move to the test line, you would get a color change because that antibody has a component that's going to react to that substrate. So again, the benefit of this is you don't have a lot of like pipetting and switching, or you don't have machinery doing that for you, but you're only getting like one sample per test, per dipstick. So there's then the flip side of that cost. Usually these lateral flow analysis systems are relatively are cheaper to construct and make. So there's a cost savings on that sense. So you may be using more tests, but they're not as expensive as per test, because you don't need these additional reagents that you would need for an ELISA, or different additional machinery you would need for an ELISA. Usually the reading for these are relatively short too. So usually you can get results within 20-30 minutes from these, but again, you would be limited in what is available commercially, unless you're working in a research lab developing new tests. Now, looking at nucleic acid-based methods, this is where we could have, there's several different methods. So we can have hybridization. This is where we would have a probe that's specific to the pathogen. They would recognize the DNA of the pathogen, and then the probe would then change color through the different steps of the technique. So this is limited in that you need to have a pathogen-specific DNA probe. So not all pathogens have these probes available. With PCR and qPCR, the benefit of that is that you can do both viral and bacterial. So for a lot of these techniques, if we're trying to do any of the culturing, that's going to be easier to do in bacterial, where viruses need a host cell, so you'd have to do tissue culture. So with our viruses, you notice I haven't really mentioned them much. So you could definitely use some of those immunological-based techniques, but you're kind of limited, and so this is where these PCR, qPCR techniques would be beneficial. With DNA fingerprinting, that can be utilized. However, you need a pure culture, so it can take 5 to 7 days in order to, from beginning to end, between culturing and then also doing the DNA fingerprinting and getting your results. The same kind of downside with whole genome sequencing and microarray or mass spec is that you need a pure culture. So if it's a mixed culture, if there's a contaminant in it, it's going to skew your data. And so you've taken all this time to get a pure culture, you think, but there's a contaminant in it, and then your data is useless. So those are huge downsides with those techniques where you need a pure culture. Where a lot of the food industry is going is looking at biosensors. So with biosensors, you're going to have your target substance, and then using a bioreceptor, which could either react with enzymes, cells, organelles, tissues, or you could utilize antibodies, antigen, nucleotides. It will end up detecting. This ends up causing a chemical signal, so then you need a transducer. You need something to read that, so aka machine. And then from that reading, you can get you can have signal amplification, and then data processing, so you can get, how much is there, how much signal did you get. Okay, so there's a number of different platforms, companies that do biosensor. So I just selected one of them to kind of highlight. No financial contributions are given to me, so it's not that I'm picking one over another because I'm benefiting from it. I just wanted you to be aware that there are different platforms. And again, just looking at one of them is this MicroSnap test. So in this, you end up having a detection device. So this is the machine that's actually going to read the sample to determine whether microbes are present. And so they have a number of different detection systems where you can detect E. coli specifically, coliforms, enterobacteria, or total viable counts. So depending upon what kits, what tests you purchase, you can detect these different microbes in it. So basically what you have is a swab that you will swab the material with. You then take that swab and put it into some enrichment material, so a second tube. I believe those blue caps those are the enrichment tubes. And then after a short incubation time, those tubes are then put into that machine and read to determine whether there's any microbes detected. So with this MicroSnap test, you can use it on raw material that's coming into a facility, at the facility, so actual equipment, to see whether your sanitation methods are being effective, so whether you have any contaminants along that processing pipeline. And then you can also test the finished product. So benefit of that, you don't need different tests at different points. The test could be used across the board. The thing I forgot to mention in that previous slide, if you view it on PowerPoint, there is a video that talks about the procedure to do it, and you'll notice that the procedure, compare that to your standard plate count where you're doing a serial dilution and then you're plating, and think about from a training personnel at a company on how to do which technique, which one would be easier to train someone to do, and definitely the MicroSnap test would be easier to do. So there might be a little bit more of a cost to it, but you would be able to run more tests in a short time. And again, a huge benefit here is if you look at this figure up on the right side, if you collect a sample, shift to starting, so you get raw material in, you test it, shift to starting, they're processing it, you're already working on, because you put that sample into that enrichment material, it's growing to the point where now you can test, and by the end of the shift you would know whether there's presence or absence of a specific microbe or what the total counts were, so that you can then release the product by the end of the shift. So again, this is extremely valuable because you would know before the product is even left whether the material that at least it started with, or if you're doing sampling during the process or the finished product, you would know whether it could be released or whether there's issues with it. The other benefit is that based off of the readings they get, so relative luminescence units, that can be worked back to the traditional colony forming units per ml or per swab. So you can have an idea of the quantitative aspect to it. So another newer method would be cell-based sensors, and this is kind of pretty cool because what they have are these cells that, based off of receptors on them, will recognize the pathogen or toxins, and then they're actually have a sensor in them. So obviously there's some alteration of these cells. So they have this sensors that if they sense that pathogen or toxin, the cell ends up lysing, and it actually releases these signaling molecules so that they can then be detected. So again, you'd have the cell would produce, potentially GFP or bioluminescence sensor, and if it detects, because individual cells are going to be more sensitive than what we can see and what a lot of these different methods allow. So if they sense a pathogen or toxin, they turn on the genes for the signaling molecules because they're tied together in cell signaling pathways, because that's how the cells have been engineered. So they then release like an enzyme or bioluminescence, and then that gets detected by these sensors. So the sensitivity levels of these are much higher than a lot of our other testing because it's simple ligand receptor binding, cell signaling pathways, gene activation, expression, so that you then can detect. So the thing to keep in mind is that there are published, approved methods for every area of food, the food industry, and so some of that regulation comes from the FDA, and some of that regulation comes from other public health organizations, and so that they have standardized methods. So as you can see, there's standardized methods for dairy products, standardized methods for shellfish. All these different foods are going to have standardized methods, and so companies need to follow these standardized methods. And that makes sure that one company is not using a less selective method so that it looks like they don't have any microbes present, where another company is opting to use a highly selective method and really making sure they're turning out high quality food. So it kind of standardizes the playing field that these companies are operating on. Okay, and part of when we look at the fermented foods, we will definitely be looking at some of these standardized operating protocols, and what is required of these companies as far as testing. Also, when we look at foodborne illness, some of the additional pieces, like we'll probably most likely with foodborne illness look at shellfish, and so what are the protocols and methods that are utilized. And so this measuring of microbes in the food will be an aspect that kind of works itself through the rest of the semester. So definitely make sure that you do understand some of these different methods, but keep in mind that some of the additional details, giving actual examples, will be provided later on in the semester when we're looking at specific types of foods. Thank you.